Hello and good evening. Welcome to the National PKU Alliance and Homology Medicines Gene Therapy and Gene Editing webinar. My name is Katrina Swenson and I'm the Project and Communications Coordinator for the National PKU Alliance. Tonight's webinar will be recorded and all attendees will be muted. If you have any questions during the webinar, please click on the chat icon and send me any questions you may have. Questions will be addressed at the end of the presentation if time allows. And it is my pleasure to introduce Christine Brown of the National PKU Alliance. Christine?
Thank you so much, Katrina. Thank you so much, everybody, for attending and learning more about gene therapy and gene editing as a possible treatment and cure for PKU. Before we get started, for those of you who don't know a lot about the National PKU Alliance, our mission is to improve the lives of individuals and families with PKU and pursue a cure. We're made up of 17 different state and regional organizations across the country that provide support on a local level. And in the last 10 years, we've invested over $2.7 million in research to better understand PKU as well as accelerate new therapies. Of that, so far we have invested $400,000 in gene therapy. If you're interested in helping us move PKU drug development forward, the most important thing you can do is join the PKU patient registry. Since we launched the registry in the beginning of 2017, we now have 719 participants. So these are PKU adults as well as parents and caregivers of PKU children from 44 states and 10 countries. And they have completed almost 5,000 surveys about their PKU. And the data that we're getting and the longitudinal data that we will have is what we need in order to accelerate these therapies. So at this time, I would like to introduce Albert Seymour. He is the Chief Scientific Officer at Homology Medicines. As you know, gene therapy is a very hot topic in many disease communities, including PKU. In fact, right now, there's more than 2,600 gene therapy clinical trials happening worldwide. And last year, the FDA approved the first of 3 gene therapies to treat cancer and a hereditary form of blindness. So Albert is going to share with us how gene editing and gene therapy could be used as a future treatment and cure for PKU. Albert?
Hi, Christine. Thank you so much for the introduction and the opportunity to speak to those of you on the phone with this webinar. So I'll be going over gene therapy and gene editing for PKU. Before I start, I'd like to give a brief introduction of Homology Medicines and our mission. We are actually developing therapies that we call genetic medicines, and it's based on a platform of different delivery vehicles that we use to deliver these genetic medicines. We as a company, our mission is to utilize these genetic medicines to deliver cures to many genetic diseases, and ideally for PKU in particular. And I'll go through some of that information as we go through the presentation. As far as that, my name is Albert Seymour. I'm the CSO, as Christine had mentioned. I'm a human geneticist by training. I got my PhD in human genetics and then went on to train at Pfizer Pharmaceuticals, where I really learned about the drug discovery and clinical development process. And then went on to Shire Pharmaceuticals, where I focused on delivering new medicines for patients with rare genetic diseases. And then in 2016, came to help really form and develop Homology Medicines into the company that it is today and deliver these therapies to patients with rare genetic diseases. So before I start and going through all this, I'd like to really highlight the role genetics play in PKU. And I want to start at a very high level, and that's the genetic material that all of us have in each one of our cells. So this genetic material is inherited through 23 chromosomes that we get from our mother and 23 that we get from our father. 22 of these 23 chromosomes are what are called autosomes, and they contain most of the genes in the genome. And then the last chromosome is either an X or a Y, and that's what determines whether you're a boy or a girl. As we look across these chromosomes contain all of the genes in the human genome, and PKU in particular, the gene that causes PKU, which is phenylalanine hydroxylase, or PAH for short, as I'll call it throughout the rest of this webinar, is maps to or resides on the 12th chromosome, chromosome 12. With these chromosomes, one of the other aspects that we know, and we've known this for a long time, is that how the PKU and the gene in PKU is inherited from parents to their children. It's inherited in what's called an autosomal recessive form, meaning that you have to have an affected allele from your mother and an affected allele from your father in order to have that disease. And so that's depicted over here on the right part of this figure, that if you have a father that has an affected allele, and we're defining this as a little r, and a normal allele as a big R, and then you have an unaffected carrier mom that also has a little r, so that's an affected allele, and the big R is unaffected. When they have children, each time they have a child, there's a 25% chance that they have a child that does not contain any of the mutant forms of the PAH gene. And so here you can see that the father or the child gets a big R from the dad and a big R from the mom. About 50% of the time, they'll have a child that carries at least one affected PKU gene and a normal allele. And this happens 50% of the time, because you can see you can have the little r from the dad and the big R from the mom, or the little r from the mom and the big R from the dad. About 25% of the time, they'll have a child with PKU, because that child that will inherit an affected allele from the mom and an affected allele from the father. And this is autosomal recessive because you have to have both of those in order to have the disease PKU. Now we move on to the history of PKU, and I want to introduce this timeline at the very beginning to just reiterate and highlight how long and the amount of research that has gone to understand this disease and what that takes, what is taken with this research in order to deliver new therapies to patients with PKU. So it was as early as 1934 was when when PKU was first diagnosed as a disease PKU. It took another 20 years from that before they realized that you had the phenylalanine that would rise in the blood, and that a way to help reduce that is to put patients on a low phenylalanine diet. So take what is elevated and just remove that from the diet. Even with that known, it still took almost another 10 years before newborn screening was introduced in the United States, and that was done in 1963. Follow that with approximately another 20 years after they were doing newborn screening and treating PKU patients with a low phenylalanine diet, and that's when their first paper came out that showed that DNA mutations in PAH, which is on chromosome 12, is linked to PKU. That was in 1986. You then go forward almost another 20 years before you have the first FDA-approved treatment. And the reason that is because you really have to understand what's the genetic cause of the disease, and once you know what the genetic cause of that disease is, what do we know about how that gene works in the body so that we can then target different molecules and different medicines to affect that. And that's what you saw in 2007. We then move to today, and you're starting to hear about the first true genetic medicines for this disease, where you're coming in and trying to replace or correct the actual gene that causes PKU, in this case, PAH. And I wanted to highlight this just because it really gives a good example of how that research has led to an understanding of the disease. And then once we have an understanding of the disease at the cell level, we can then start to develop different therapies for that particular disease. I then want to move to what it takes once you understand that biology, and what are the different parts of going from an idea in whether it's in a university or in an industry lab, to actually take that through the process before these medicines are available to patients. So early laboratory research, you may start to understand and discuss and do work in the lab, trying to understand if we make PAH in a different way, how much of that PAH do we need to make to have a therapeutic effect? Once you get a good handle on that, you can then move to what we call preclinical development. And that is when you start on two fronts. One, working in the laboratory to continue and get more information around that protein. How much do you need in a cell? How much can you deliver to different models? And how often and how durable can you have that effect? While that's ongoing, you'll see at the top, there's also this line we call manufacturing. This is also a very important component of bringing these gene therapy and gene editing programs to patients. We need to be able to make the therapy consistently and safely over time. And that starts as early as preclinical development and then continues through that process. So once we come out of the laboratory work in preclinical development after testing in different models, we then move into clinical trials. And that's for the first time that we can take these therapies that have been demonstrated to be safe as well as efficacious in preclinical models to then start testing them in the clinic. And this is when we first directly start to interact with many of the doctors that PKU patients see. And that's how we start to interact with them, those doctors, to help design the clinical trial, what would be the best approach to test a different therapy in patients with PKU. This typically starts in a small trial where we look and test this initially to see how well tolerated is the therapy, and then also how efficacious is that therapy in a small number of patients. If that looks good, we then move to testing it in more patients so that we can get a better understanding of the safety and the efficacy of the gene therapy or gene editing approach. All along on the manufacturing side, we're continuing to learn how to make the gene product, the gene therapy product, safely and consistently so that we can take that whole package. So all of the information that we generate from discovery through those clinical trials, we then present to the regulatory agencies. So in the United States, it's the Food and Drug Administration. They'll then take our data and our information, and they will review that and determine whether the therapy for patients is efficacious, whether it's safe, and also whether or not we as a company can manufacture and produce that therapy so that we can then not make it available to patients and always have that therapy available to patients. And it's at that point that it comes back around to the doctors that treat PKU patients to then make this available to the larger PKU population. It is a rare disease. Sometimes this path can be quicker in a rare disease, but again, it all depends on the type of data and the type of information that you're generating. So that's why you'll see these big ranges in years. But I wanted to highlight this just to give a sense of the path and the length of time it can take to go from an idea to having this as a medicine that your doctor can prescribe and use to treat PKU patients. Now that we've gone through all of that, I want to come back to PKU at the genetic level. And I want to walk through this because this is extremely important as a way to understand what's happening in the liver and in particular the cells in the liver, because this is important for the therapies that are available today, as well as designing drugs for genetic medicines. So if you look at PKU at the genetic level, the enzyme itself is expressed and works primarily in the liver. So if you look at this patient, if you look in that liver, if you then highlight and focus in on the cells of that liver, for someone that does not have PKU, their liver cells will have their chromosome 12, and then within that cell, there's a factory. That factory is called a nucleus. And coming off that chromosome 12, that's where the gene for PAH is, and it runs through that cell machinery to make a functional enzyme, an enzyme that can break down and metabolize phenylalanine normally. For a patient with PKU, if you look in the liver of their cells, it's the same process. You have chromosome 12 that sits in this factory within the cell called the nucleus. That DNA or the gene from that sits on chromosome 12 has a mutation in it. The factory is still working, but the product that comes out of that factory is a protein that does not work. And because that protein does not work, then it tries to metabolize phenylalanine and break down phenylalanine, but it doesn't. And I'll go through a little bit more details of why that's important. So if you go on the next slide, Again, coming back to a functional enzyme. So in a person that does not have PKU, chromosome 12 is making that protein. It's going through the factory. It's making a functional PAH, or phenylalanine hydroxylase. If you highlight and look in the middle here, you'll then see that as you eat protein comes in, it starts to break down to phenylalanine. Phenylalanine hydroxylase is in your liver cells. It will see that phenylalanine, and it will start to break it down into a product called tyrosine. And then tyrosine can be used to produce many other different types of proteins. Melanin, which is responsible for skin color, hair color, or what are called neurotransmitters. These are different types of molecules that can go into the brain that can help your brain cells communicate to one another. So in this population, you have PAH, you don't get phenylalanine buildup. If phenylalanine converts to tyrosine, and then you produce these downstream products. The next slide shows what happens when you do have a mutation in PAH. So the mutation in PAH on chromosome 12, again, that factory thinks it's going through, it's making what it thinks is an active protein, but it's not until the part that product of that factory comes out that it's non-functional, it doesn't work. And so because this doesn't work, you have protein comes in, that protein gets broken down into phenylalanine, it's an amino acid, and then it's almost like a dam builds up. It's trying to come into the cells, and these cells no longer can break it down. So because that can't break down, you start to see phenylalanine build up and build up and build up. And as that phenylalanine builds up in the blood and the brain, this is what leads to all of the complications in PKU patients. And that requires those patients then to go on a phenylalanine restricted diet, sometimes tyrosine supplementation, and then can lead to some of the effects that PKU patients feel and have. So if you go to the next slide, as you go through that and really understanding how these factories work, that information took decades of research to understand it at that level. And because of that research, doctors and pharmaceutical companies and universities could all look at that and say, how best to treat these patients. So there are 3 treatments that are currently available. The standard of care is the low phenylalanine diet that I'm sure all of you are familiar with. This is obviously a daily treatment, and this is to lower the amount of phenylalanine that's going on. There's also a drug called Kuvan. Kuvan actually is called BH4. This is a cofactor. Think of it like a partner. So for the enzyme PAH, it needs its partner in order for it to be active. In the absence of that partner, it cannot be active. So with Kuvan, you have this partner that helps it work. That is also a daily treatment. And then obviously more recently is a drug called Palynziq. This is a little bit different. Palynziq is a daily treatment. It's injectable, and it's using an enzyme called phenylalanine lyase, and that works in the blood. And I'll go through and describe each one of these mechanisms because I think it's really important to understand these as it relates to thinking about genetic medicines for PKU. So if you go to the next slide, this just breaks it down a little bit more. So obviously with the phenylalanine restricted diet, you have this dam build up because phenylalanine cannot be broken down because the PAH is not functional. So one way to avoid that buildup of phenylalanine is to lower the amount of phenylalanine that comes into the system. And so thus you have your phenylalanine restricted diet. For Kuvan, I was talking about this partner. So it's for patients that have some active PAH, some residual active, and then you have Kuvan that works as a partner to help give that even a little bit more activity. So that then you can take some of that phenylalanine and metabolize it and break it down to tyrosine and the other byproducts that we spoke about earlier. Palynziq is a little bit different. It targets phenylalanine. It works in the blood. So the phenylalanine levels get very high in the blood. When you inject this enzyme, phenylalanine lyase, rides around in the bloodstream and sees and detects phenylalanine in the bloodstream. And once it binds that, it breaks it down. What it does is it breaks it down into different products, tranexamic acid and ammonia. So it takes phenylalanine and gets rid of it through this pathway as opposed to this. So the functional enzyme PAH is still there, that doesn't work, but you're lowering phenylalanine in the blood via this pathway. With all of those, that's why these new approaches to treat and potentially cure PKU are focused on correcting the PKU at the gene level. So we know that PAH is the causative gene, and so there are really two approaches. More traditionally is gene therapy. This is an approach that's been in development for several decades now. We're really starting to see a lot of these come forward in many different diseases. Gene therapy, what it does is it adds a functional copy of the gene. So it takes a working copy of PAH and puts it into the liver cell. It does not integrate into your chromosomes. So it will sit inside this factory, but it kind of works outside the chromosomes. This is an approach that's most suitable for any organs that don't divide very rapidly. So whether it's in a liver in an adult or in different brain cells, because you want this to sit inside that factory and stay there. It's a potential for a one-time treatment, and I'll walk through several examples of that. Gene editing, on the other hand, again, it inserts a functional gene, but what makes it different than gene therapy is it actually takes that gene and it does go into your chromosome, and it would be focused in on a region such as chromosome 12 where the PAH gene lies, and it integrates into that genome. And this is very important, particularly for organs that are dividing rapidly, such as a child during development. So their liver is going to grow, and as that liver grows, you want to have the DNA fixed as they're little, and then each time those cells grow, that liver grows, the liver cells that grow over time are also fixed. And again, this has the potential for a one-time treatment. So now I'm going to walk through a little bit about gene therapy, and then I'll talk about gene editing. I want to divide the two because there are some distinct differences between the two approaches. So gene therapy, one of the things you have to do is you have to have the ability to deliver this genetic medicine. So we want to take this functional copy of the gene and we want it to go to the right part of the body, and in PKU, this is the liver, and we want it to go to the right part of the liver cell. In order to do that, we have to come up with different ways to deliver these. There are many different approaches. The one I'm going to highlight is called AAV, or adeno-associated virus. But there are other approaches that I'm not going to talk about today, but obviously I'm happy if you have any questions about any of these other approaches, more than happy to address those questions. AAVs are very important because it's a delivery vehicle that we can manufacture, but more importantly, they've never been associated with any human or animal disease. You may see the word virus in that, but they're non-pathogenic, meaning they don't cause the disease that we know about. They sit inside humans for a long time, and actually many humans have them in them. They're able to deliver two key organs, such as the liver. And what I mean by that is that we can put these into your bloodstream, and they travel to your liver cells. The other important thing about this type of approach is that they've been now used in more than 200 clinical trials worldwide. So there's a very big safety database. There's also a really good understanding about how these AAVs can be used to deliver these type of gene therapy approaches for genetic medicines. On the next slide, this is how I'm going to go into a little bit more detail and explain actually what those gene therapies and AAVs are doing when they go into the body. So if you look on the left, if we take a patient, this is a female patient who has PKU, you can see it, we want this to go to her liver. We can take these, we put them into her bloodstream, and these go to the liver. They're very good at going to the liver. Once they're in the liver, they can go into the liver cell, and here you can depict what that gene therapy looks like. It's inside that gene therapy is what really is important. So this blue part here, that represents the functional copy, a good copy of PAH. And the green part is there. That's important for once this gets into the cell, it has to go into this factory of the cell called the nucleus. And you'll see it forms these little circles. And it's these circles that can then take advantage of that factory. It almost robs some of the machinery that this factory uses to turn on the good copy of the gene. And that results in a good copy of PAH. One thing I want to highlight is that the bad copy of PAH is still in that cell. We're not going into the chromosomes. Now, what does that do once you have it inside the cell? So this allows, once you deliver it into the liver cell, you have this circle. It's taking advantage of the machinery and all of the elements of this factory inside the cell to turn on PAH. It makes this copy of PAH, and it can go inside the cell. Now, if you look in the center part, protein comes in. It's broken down into phenylalanine. That phenylalanine then comes into the liver cell, and it sees all of these functional copies of PAH. And because they're there, instead of being blocked and building up as an edema, you have these functional copies of PAH that then start to work in taking phenylalanine and turning it into tyrosine, and then all the other byproducts of tyrosine, melanin, as well as the neurotransmitters we talked about. When you do this, you start to see that phenylalanine reduces. Then you don't see this phenylalanine build up over time. It then restores the natural biochemical pathway, meaning that phenylalanine is reduced because this good copy of the gene is converting it to tyrosine. This is how that gene therapy works in that liver cell. One thing I will highlight here too is I was talking about slowly dividing cells. You can see that this is a circle. If that cell divides, one cell will have that, the other cell won't. And that's why it's important from a gene therapy perspective to be in cells that don't divide very rapidly. And I'm going to highlight how gene editing that we can target for much younger patients can overcome that particular challenge that gene therapy poses. I just want to end on the gene therapy part to say, and Christine actually mentioned this earlier, there is one that's already available to patients. There's a company called Spark Therapeutics. They have a medicine called Luxturna, and what Luxturna is doing is they're using these AAVs, and it delivers a functional copy of another enzyme. This enzyme is called RPE65, but it delivers it to eye cells. And what happens in these patients that get this, they go from not being able to see to being able to see. So it's a tremendous benefit to these kids that have this inherited eye disease. And so all in all, if you take all of the gene therapy trials using all the delivery approaches in monogenic single gene disorders worldwide, there are almost 300 of these trials that are ongoing. So what you're going to start to see are regulatory approval for many of these this year that ultimately lead to therapies for patients with several of these diseases. I'm going to shift gears now to gene editing. And before I start, I just wanted to highlight that if you go out and Google gene editing, you may hear about CRISPRs and other things like bringing woolly mammoths back or engineering the perfect baby. This is not what we're talking about. What we'll be talking about are scientists and companies that are using tools to correct DNA in the cells of a person. We're not correcting those cells that are then passed down to your children. It's only correcting the defect or the mutation in the person that we're treating. So how do we do that? In order to do this with gene editing, and gene editing is a newer technology than gene therapy, you have to have the tools that allow you to go in and engineer the genome and the chromosome. You have to have the tools that can go in and allow you to put a gene in or turn off a gene in that chromosome. There's several tools that are available that are out there. You've probably heard about CRISPR. CRISPR is what's called it's CRISPR-Cas9. It is what's called a nuclease. A nuclease is just the name for an enzyme that cuts DNA. So nuclei is for the nuclear part, which is DNA, and the ase part is because it's an active enzyme. And so because these nucleases like to come in and cut DNA, and so you can use them to specifically go into a region in the genome, find that region in your genome, say it's near the PAH, and then you cut it. It cuts the DNA in half. And what that does, that then tells the cell that there's damage to the DNA, and it evokes the DNA repair process. And so it puts inside this factory the machines to fix that. And that's what you can use to insert something in during that fixing process. There's another tool, it's called homologous recombination. This is also another way that you can insert genes into the chromosome. It is a natural process. All of us are undergoing homologous recombination in our cells right now. What this does is this uses long arms, and we call them homology arms. And these arms are long stretches of DNA that are almost like zip codes. It'll take the gene of interest to a zip code in your genome, say chromosome 12 for phenylalanine hydroxylase, and it will put something there in that cell. So those are the tools. With those tools, you also have to have the ability to deliver those tools to the right place in the body. And so you'll notice here that AAVs are on there as well. So we can use AAVs for gene editing to deliver either nucleases or this homologous recombination approach and take advantage of those properties to get these genetic medicines to the cells. I'm not going to talk about other approaches, but there are other approaches to deliver. The delivery approaches, all in all, are really meant to protect the genetic medicine from your bloodstream. You have to protect it. Your bloodstream is very good at getting rid of DNA that's not supposed to be there. So in a sense, it's a way to hide it and then take it to where it needs to go. And then once it's inside the cell, then it's safe to come out. So now for gene editing, this is how that process works. And I'll focus on homologous recombination. For one reason, that's the approach that we're using to bring an editing approach to children with PKU. And this is something that's being developed right now. So again, here you can see on the left to deliver, this would be packaged and put into the bloodstream. You can see here's a picture of a child with PKU. That then goes through the child's bloodstream, gets into the liver. And then once it's in the liver, I'll move over here. You can see we still have the same delivery approach, which is AV. You'll have the blue part, which is PAH. But notice we don't have that green part. That's the part that turns on the gene. We have these zip codes called homology arms. And that's a way to almost think about it like the post person is delivering it to a region on your chromosome 12, so that it comes in, it finds where that mutated PAH gene is. It undergoes this process in the factory of the cell, which is a nucleus, and it takes out the bad copy and it puts in this good copy. Once that good copy is in there, the rest of the factory continues to work, and now it makes a functional phenylalanine hydroxylase or PAH gene. So why is that important, and how would that work in PKU? So if you go to the next slide, this is a depiction of that same cell working through the factory. It's corrected in the genome, so it's on chromosome 12 within the PAH gene. It's making a functional copy of the PAH gene. Now protein comes in, phenylalanine is broken down, and then within that, because of this fix, we have an active form of PAH or phenylalanine hydroxylase that then allows, instead of phenylalanine building up, it starts to break that phenylalanine down into tyrosine and its natural substrates, melanin and neurotransmitters. So similar to gene therapy, it's the normal biochemical process of putting PAH in the liver and breaking down phenylalanine in the liver, so you don't get phenylalanine buildup. It converts it to tyrosine and produces these neurotransmitters. What's different from gene therapy is it's inside the DNA. So as that cell starts to grow and that child's liver starts to grow, this carries on during the growth of that particular liver. These programs, because it's a new technology, are about 1 or 2 years behind. If you remember that pathway to getting medicines to patients, it's about 1 to 2 years behind. So whereas the gene therapy programs are starting to move to clinical trials, this is in that part called preclinical development, where we're going through and really testing to understand how effective and how safe this can be in the models. This gene editing is also already in the clinic and available to patients with other rare genetic disorders. And so there's a company called Sangamo. They're running the first, you'll see on here it says in vivo gene editing. What that means is it's the first trial where they're adding this into the bloodstream. It goes to the liver and it's making a cut in the DNA and then putting in a good copy of the gene that causes Hunter syndrome. And you can see the picture of the patient on the upper right who has received this therapy from them. What you're seeing now is that there are many other gene editing trials by several other companies. You'll be hearing about these more and more over the next coming years as this approach tries to target these different disease types, so that we'll have a toolbox of genetic approaches to target slowly dividing or non-dividing cells, as well as dividing cells and pediatric type populations. So I went through a lot. I know I covered quite a bit in gene therapy and gene editing. There are many resources available if you have any questions or want more information on these approaches. Obviously, through Christine and the National PKU Alliance has quite a bit of information. The National Organization for Rare Disorders has a lot. The American Society of Gene and Cell Therapy focuses on the development and discovery of gene therapy and gene editing medicines. And then finally, the U.S. Food and Drug Administration, if you have questions about what kind of trials, what does it take to run a trial, what does that look like, the FDA actually has really informative information for not only people in industry, but anyone that's interested in that. And then finally, any questions about them in particular, feel free to reach out through email at patientadvocacy@homologymedicines.com, and we're more than happy to respond to any of those questions that come in. So with that, I want to thank you for your attention, and I'm also open to any questions that anybody may have.
Thank you, Albert. This is Katrina. I do have a question that was sent via chat through Danielle, and she asked, after gene therapy, is there a different medication or a PKU person would have to take for any reason? For example, like a transplant patient needs to take medication to prevent rejection or combat immunosuppressants for the remainder of their life.
Yeah, so that's a great question, Danielle. Thank you for the question. The way these gene medicines are being developed is that it would be a one-time administration, and that one-time administration would add that functional, say, PAH gene back into the liver. Once that's established, the thought or the idea is that you would not need any additional medicines, particularly like those would needed for transplant patients. The ideal would be that it would fix the underlying genetic cause, and you would be able to go on without those additional medicines for the rest of your life.
Thank you, Albert. It doesn't look like we have any more questions at this time. As Albert mentioned, you can reach out to Homology Medicines, or you can also reach out to myself, Katrina Swenson, at katrina.swenson@npkua.org. And we hope that you enjoyed tonight's webinar and found it very informative. As I mentioned, any questions you may have, please feel free to contact me. This recording will be available on Tuesday, and I will make sure to email a link via an e-blast. And I just want to thank Albert from Homology Medicines and everyone for attending this webinar. Thank you again, Albert.
Yes, thank you, Katrina.
Have a good evening, everybody. Thank you.